Okay, excellent, got it. So Dr. Michele Buzon is a professor in the Department of Anthropology at Purdue University with a research focus in bioarchaeology, paleopathology, cultural contact, and environmental stressors. Dr. Buzon has been conducting bioarchaeological research at the site of Tombos in the Third Cataract region along the Nile since the year 2000. And this project in particular examines interactions between Egyptian and Nubian populations. And so, without further ado, I'll pass the floor to Dr. Michele Buzon.
All right, thank you, Robert. Thank you very much for inviting me to this seminar. Let me share my screen. Does that look okay for you?
Looks good.
Okay. All right. So today I'm going to talk about variability in isotope values in the Nile Valley and using this method to identify first-generation immigrants in archaeological samples. Make sure. All right. So much of what I'll present today comes from the National Science Foundation project that we are currently completing, as well as the work that the team has been doing on this topic over the last 20 years since I started strontium isotope research in the region around 2004 during a postdoctoral fellowship at the University of Alberta. For this NSF project, we propose to obtain additional strontium isotope analyses of faunal, sediment, human remains, and plants, in particular those that are well characterized in relation to their age, and compare these to their present day equivalents. And the main goal here is then to develop a temporal evolution or stratigraphy of the bioavailable strontium isotope signature of the Nile River Valley. In addition, our goal is to combine the new strontium isotope data with isotope analyses of lead and neodymium samples to be investigated, since the latter two isotope systems will provide some additional insights into provenience and possible changes over time within the Nile River Valley. So I have been doing this project with Tony Simonetti when I started my postdoc at University of Alberta, and we've published a number of papers over the years. I started this work as a way to more fully explore the Tombos community, given that the archaeological and skeletal evidence at Tombos suggested that both immigrants and locals were present. And when I finished my time in Alberta, I went to Purdue University, and I intended to maintain this collaboration with Tony at Alberta because we had, a great working relationship. And fortunately, 1 year later, Tony ended up at University of Notre Dame, and that's just a couple of hours from Purdue. So we have been able to work quite closely together, sharing samples, doing lab work, those sorts of things. So in addition to Tony, my main collaborator on this project, Loretta Corcoran worked as a postdoc with Tony on this project. Carrie Gilbo worked with me here at Purdue as a research assistant. And this is the result of work that a number of people have been doing, including Sarah Schrader, who's here with us today, working on strontium from sites in the region as well. Dr. Maha Kordofani from University of Khartoum worked with me on identifying plants to use in this study as well. So over the past few decades, the use of strontium isotope compositions have made an important contribution in understanding the provenance of materials in archaeology and the interactions and migrations of ancient civilizations throughout the globe. The strontium ratios of biological and geological materials vary as a function of geological provinces. So they're dependent on the age and mineralogical makeup. There are areas that are characterized by older bedrock with a higher proportion of minerals containing high rubidium to strontium ratio, such as mica, yield higher strontium 87 to strontium 86 ratios compared to regions that contain younger rocks with lower rubidium to strontium ratios. The strontium ratios are then transferred into the hydrosphere and the ecosphere through weathering. So animals record strontium isotope compositions from their environment and diet, and this signature is incorporated into their organic and skeletal tissues. Organic tissues that grow continuously, such as hair, teeth, bones, tusks, They then record temporal variations in the strontium isotope ratios, which can then be used to decipher migration patterns of ancient individuals or groups. With regard to the Nile Valley Basin, previous studies have acknowledged that in relation to the fluvial record, this region of the Earth is well suited for provenance studies based on strontium isotope compositions, because the upper reaches of the Nile's major tributaries traverse distinct geological and hydrological environments. The source of the White Nile River originates in the ancient shield rocks characterized by high strontium ratios, whereas the rivers of the Blue Nile and the Atbara flow over much younger Cenozoic volcanic terrains of the Ethiopian uplands with much lower strontium ratios. At Khartoum, the confluence of the Blue and White Niles, the isotopic signature of river sediment in the Desert Nile and the Delta has been interpreted as a weighted mean average of the isotopic signatures of end member rocks eroded from the upstream drainage basins. The detailed strontium isotope maps are useful to a certain extent, but they're somewhat limited in use for archaeological samples, as bioavailable strontium does not need to have the same isotopic ratio as the stationary strontium. Modern reference samples of soil, plants, and animals can be used, but also can be sometimes problematic because of the use of carbonates and mineral fertilizers, as well as imported feed. So in using this analyses, strontium isotope analyses, to understand archaeological populations, it's best to use skeletal remains of archaeological herbivores as a proxy assessment of local bioavailable strontium. So small local animals whose diets have been sort of in a small range, and they've been shown to reflect average regional values. That's really what's ideal here, small animals. However, I certainly have encountered issues in finding small animals in the archaeological record, and many colleagues have as well. Many of us do find animals such as sheep, goats, cattle, but they can be limited in usefulness as well because they have grazing patterns that may cross geological regions. Archaeological fauna can have some biases as well. They're susceptible to diagenetic contamination. So there's a variety of challenges in determining the local bioavailable strontium, with people using both archaeological and modern, depending on what's available. So many analyses use the method established a couple decades ago by Doug Price to determine local strontium signature, which is the mean value of these animals, whether it be modern or archaeological, plus or minus 2 standard deviations. This is, a arbitrary range. So other studies who have sort of gone about this a different way, maybe didn't have faunal samples, have used methods to identify statistical outliers that deviate from the normal distribution. Statistical identification is one way of going about this, but it is challenged by our often small sample sizes. I myself, as I've mentioned, have had difficulties gathering archaeological fauna in the Tombos region from the sites available, and so have often relied on modern. We asked local kids, as you see pictured here in the left, to gather some mice and rats, and that's what we've used to determine local range at Tombos. So it has been suggested that global climate changes throughout the Holocene influenced Nile Valley drainage, with aeolian sediments having a major impact to the valley floor sedimentation. And as a result of this, it's proposed that the temporal strontium isotope signatures and bioarchaeological approaches that assess population mobility using this method may be impacted. The support for this idea relates to the concept that aeolian windborne dust may have been ingested by Egyptians and Nubians, resulting in a progressive increase in their strontium isotope values. And researchers point to the extensive dental wear in the Nile Valley populations to corroborate this idea. However, we think that there are several inconsistencies with this hypothesis. In relation to the ingestion of aeolian dust, it is composed primarily of sand, which is quartz, and clay. And the former is well known to be devoid of trace elements, including strontium, due to its compact crystal structure, which resists elemental substitution. The strontium isotopic composition of clay in the Nile River region has yet to be determined and depends on the composition of the source rock and the time of weathering. To our knowledge, investigations into the manner in which clay is metabolized and incorporated into tooth enamel are lacking. In addition, though, tooth wear is more likely attributed to grit emanating from grinding stones. Grain during the past was processed using stone, quern, and mortar, which can introduce grit into grain, accelerating tooth wear. The grinding vessels that were used were made of quartz generally, and as stated above, quartz doesn't contain much strontium, so its ingestion would not perturb an individual's strontium isotope composition. So much of what I'm going to be talking about today and the results I'll be reporting have just come out in this in-press article that's now available in early view in Bioarchaeology International. So here's my first set of data coming from this NSF project. So this chart illustrates the strontium isotope compositions compiled from published samples of human enamel, faunal, and sediment from the Nile Valley, along with new data obtained for this study, including modern plant samples. These are the modern plant samples here along the edge. And strontium isotope values are plotted against their corresponding reported ages between approximately 7,000 years before present to present day. And as demonstrated here, the ratios have remained relatively constant through this time period, with no increases in strontium isotope values with decreasing age, as suggested by the aeolian dust hypothesis. Additionally, human tooth enamel samples in black, see if I can get my marker working. There we go. So the black represent the human tooth enamel samples and the brown represent the faunal samples. So the human tooth enamel samples in black overlap with the corresponding faunal samples in brown. And again, these are the modern plant samples over here. In order to test if radiogenic aeolian-based strontium was gradually incorporated into plants and then transferred into bioavailable strontium to humans, modern plant samples were analyzed. This time-dependent process of atmospheric bioavailable strontium accumulation is evaluated by comparing strontium isotope compositions in plants with different root systems, which vary in response to water availability and plant growth. So for example, a global survey of plant rooting depths indicates that 44% of grass roots are concentrated in the top 10 centimeters of soil, compared with 21% in shrubs. So it is generally assumed that the contribution of bioavailable atmospheric strontium gradually decreases with soil depth. So we collected plants from a number of sites, including Tombos, Wadi Halfa, and Aubrey, and colleagues contributed plants from El Kuru and Dongla. These present-day plants, as seen in the earlier chart, overlap with the older human and faunal samples. Root depth also does not show any differentiation. It takes tens of thousands of years for trees and plants to acquire the strontium isotope signature of aeolian dust. Strontium isotope fingerprint of aeolian dust becomes a significant factor only when carbonate sources are being weathered. So if the aeolian dust within the Nile Valley consists of mainly weathered silicate material, then the amount of bioavailable strontium for humans and plants is limited. You can see here in the shallow, medium, and deep root samples, they overlap quite a bit. There's still some more work and interpretation analyses we can do in looking at the variability by site and root, and that will be the subject of some continued analyses. So with the climatic shift to more arid conditions over the past 1,500 years, the change in bioavailable calcium and strontium for humans and plants would not be instantaneous. So the resultant strontium isotope composition of bioavailable strontium in the Nile Valley will reflect the mixing of two components and a mass balance calculation. It wouldn't consist solely of aeolian source completely masking the strontium from local sources. So we'll try to sort of sort this out more as we look at more plant samples. So I'm going to move on to discussing some patterns found in the human samples. Some of these results can be found in another recent paper, also available on Early View in Bioarchaeology International. So of interest to many here is the usefulness of strontium isotope analysis as a way to distinguish locals from those who spent time during their tooth development of childhood in another location. So these published human values are ranged from north to south, from north to south in the Nile Valley. And generally, you can see that there is a decrease in values from north to south. However, it is clear that there is much variability. So for instance, at Hannock, at the Third Cataract, we have a large range of values. The sample size is quite small, but they are quite variable. It should be noted here that all of these samples included in this chart aren't considered to be locals. It is still sort of a work in progress in understanding which ones, match the local range and some different interpretations here. So I will talk briefly about the work we've been doing at Tombos, and you can see here in the Tombos data that there are several individuals that are quite a bit above the local range. So what I think is essential in interpretations of isotope data is that they're done within the context of other archaeological information, understanding the biases of this method. So with the overlap in sites, it's important to question if those outside the local range could in fact be immigrants as well. So here in the Tombos data, the red box represents the local range, primarily derived from modern rodents, a few archaeological fauna, and some sediment values as well. The archaeological data at Tombos indicate that Egyptian immigrants founded this colonial town. And we find that those with non-local strontium isotope values are found in tomb styles and body positions associated with Egyptian practices. The more local tomb style, the tumulus grave, only has individuals that match the local range. Also, burials in the flexed body position, more local to this Kerma area, are also all isotopically local in their strontium values. All of the non-local values come from individuals buried in the New Kingdom colonial period. None of the non-locals date to after the New Kingdom. And a considerable number of non-locals are found in the chamber tombs. You can see here the 31% in the chamber tombs, which we have designated as middle class. This is also reflected in the cranial morphology found in the people in these chamber tombs as well. The pyramid and chapel tombs, and this here, have comparatively few non-locals. It should be noted that the logistical difficulties prevented full excavation of most of these tombs. We rarely reach the chambers and thus have a biased sample and may not be including those likely elite individuals who may be more likely to be immigrants. Both males and females, as well as children, are included in the non-local group, suggesting that families may have relocated to Tombos. I think it's important to see that there are some low values here, and some of these low values may be reflective of individuals who come from other sites upstream of Tombos, and that's something we still have to sort of figure out. So just briefly to finish up, we have used lead and neodymium to look at the relationship with provenance and also the relationship with strontium. So lead has been used in a few different places. One of the major issues with lead, though, is that it is found in very low amounts, and so it can be difficult to use in human samples. Of interest for this study is that none of the lead isotope data, especially in the human tooth enamel, overlap with the Saharan dust values. So here, the red are the Saharan dust values, and they are not overlapping here with the human values. We do find, though, that lead is useful in identifying diagenetic alterations. So samples with elevated lead contents above 3 ppm have lead isotope ratios that reflect diagenetic changes. This is particularly evident in the samples from El Kuru, a Christian period cemetery, impacted by the site's proximal location to several wadis that have rendered it prone to flooding events in the past. We also looked at neodymium, which has been used not very often, but is starting to be used more and more in human provenance studies. The strontium neodymium isotope composition for modern day soil from Nuri is less radiogenic compared to the older faunal samples from the same site. And there's minimal overlap between the neodymium strontium isotope signatures for human samples and those recorded for the North African dust and Red Sea hills. So you can see the plus the X is the North African dust, the dash is the Red Sea hills, and you can see that the human samples don't overlap a lot with those sources. So, as this recent paper cautions, the expansion of isotope analysis has transformed the study of past migration and mobility, but there are dangers in researchers using isotope data without a full understanding of the challenging issues. Clear-cut answers are often desired, but it's often impossible with the limitations we're considering. So the use of multiple isotope proxies may be an answer to this. And while we have used strontium and lead and neodymium to look at possible changes over time, and I think that climatic shifts have not affected these isotope systems, we certainly need more data to evaluate the usefulness in identifying known locals, especially where there's so much overlap. So the next steps will be more samples from Tombos or possibly other sites where we can have strontium, neodymium, and lead in order to look at how they all relate together. So thank you very much for listening, and thank you to all of my collaborators who helped with this project. Of course, Sudan is in our hearts and minds as we watch what's happening and hope for peace very soon. All right, I will stop sharing my screen.
Okay, wonderful. Thank you very much. That was a great presentation. We will keep the train moving, and we'll move next to the presentation by Dr. Sarah Schrader. Dr. Sarah Schrader is an assistant professor in osteoarchaeology and head of the Laboratory for Human Osteoarchaeology at the Faculty of Archaeology at Leiden University. Dr. Schrader's research focuses in particular on the reconstruction of daily activities, namely through musculoskeletal attachment sites, as well as stable isotope analyses, helping to address questions of workload, social identity, and human agency in the past. Dr. Schrader is currently excavating the site of Abu Fatima, an ancient Nubian cemetery near the third cataract of the Nile. So thank you for joining us today, and please take it away.
Thank you, Robert. Let me attempt to share my screen. Oops. Minimize all of you. Okay, Robert, can you confirm that my slides are changing and that you see my screen?
It would help if my volume was on. It looked good. The flags came and went as you cursored through, so I think we're all good to go.
Perfect, fantastic, thank you. Okay, I can't see any of you, but I will do my very best. So thank you so much for inviting me. This is really fantastic, a great lecture series. I'm very excited to tell you a bit about some isotopic research that I've done recently. And as you guys probably saw, and as Robert and Michele already stated, of course, my thoughts are with Sudan right now. We, of course, support our colleagues and friends and are in solidarity with them during this difficult time. And also, I just wanted to quickly note that there will be images of skeletal remains in my presentation, just in case anybody has any ethical issues with that. So I've organized the talk today into 4 sections: dietary isotopes, a bit about Bronze Age Nubia, although I won't spend too much time on this because I think a lot of us are probably familiar with this, Kushite diet, kind of like a hypothetical Kushite diet, and then go on to some of the data that I have, some future directions and recommendations that I might be able to make to some fellow researchers. Okay. So very briefly, the ratio of isotopic elements, particularly carbon and nitrogen, have been shown to be successful approaches to dietary reconstruction in the past. In C3 plants, stomata are continuously open, which facilitates evaporation of the lighter isotope C12 and results in enrichment of C13. Therefore, the values of C3 foods, sorry, the font is a bit messed up here, but the values are approximately -35 to -20 parts per million. So in the Nile Valley, this can include, but is not limited to, foods like emmer wheat, barley, tubers, nuts, beans, and a lot of fruits and vegetables. And this is next to C4 plants, which are adapted to hot and arid environments. So their stomata remain closed and thus retain those limited water resources. So this results in little to no evaporation of C13, and thus usually an accepted range is around -14 to -9 parts per million. So examples of Nile Valley C4 plants include sorghum and millet. Okay, so for nitrogen, nitrogen isotopes can elucidate what types of proteins were consumed. So for example, when a source of protein is eaten, nitrogen from said protein is then absorbed into the consumer. And this can create a chain reaction with ever increasing nitrogen levels, which is also referred to as trophic level. So in this way, we're able to very broadly discern if an individual was eating sometimes more or less proteins and perhaps different types of proteins. So I have here the example of a leaf gets eaten by a cow, which then absorbs the nitrogen from the leaf, and then a hyena might hypothetically eat a cow, and then it absorbs both the nitrogen from the cow as well as the leaf. So it creates a chain reaction of nitrogen. So a quick note about bone turnover. So typically bone, either collagen or carbonate, is used to assess dietary patterns using these dietary isotopes. But it's important to remember that bone remodels relatively slowly, taking anywhere between 3 and 20 years to completely remodel. So isotope analysis on bone provides a very general idea of foods that were consumed over years or decades before death. So this perspective is very important, even if we're looking at decades before death, because it does reflect foods consumed by actual individuals. We're not looking at on a population level, but we're actually looking at the people who consumed these foods. So even if it is a very general overview of years or decades, it's still very important. Okay, so a few notes on Bronze Age Nubia, also referred to as Kerma, based on the capital city and type site, but also, of course, referred to as Kush, or the first Kushite state. It was a strong and independent kingdom that threatened Egypt on several occasions. It was an agro-pastoral society, and some have even argued a pastoral state. There was no writing associated with ancient Nubia. But nevertheless, we know from the archaeological evidence that it was of course, very strong and independent. There was a unique religious system that we don't know much about, partially because of this lack of writing, but we do have preserved religious structures such as the one that's pictured here, which is the Defufa, which is a massive mud brick structure that no doubt held religious significance. So it's typically divided into 3 periods: the ancient Kerma, middle Kerma, and classic Kerma periods, although more recent research has shown that there was also a pre-Kerma component to this. And then also, of course, life continued after the classic Kerma period during the colonization period, when Egypt came down and conquered Kerma. But life didn't stop, and Michele's research speaks to this. There were these very unique communities that sprang up, such as Tombos, that kind of encouraged this coexistence where both Nubians and Egyptians were living side by side and interacted biologically and socially. So yeah, the story, of course, continues for Kerma. Okay, so moving on to just a, maybe hypothetical Kushite diet, what we might expect from the isotope analysis that we're doing. So funerary offerings, artistic depictions, written documents from Egypt, as well as archaeological, zooarchaeological, and archaeobotanical remains have suggested that some of the foods that may have existed during this time were breads, palm dates, pomegranates, melons, figs, cucumbers, leeks, cabbage, onions, beans, lentils, honey, goat, pig, cattle, and fish were in theory available to both Egyptians and Nubians. One thing that potentially might be, we might expect to be more pronounced in Nubians is, my computer is frozen. There we go. Cattle. So there is, a theory that a cattle cult existed within ancient Kerma culture. And this is partially because of the large number of bucrania or cattle skulls that were provided as offerings, presumably to the rulers of Kush. So in one middle Kerma period context, we have as many as 5,000 bucrania that were left as offerings around the grave of the ruler of Kerma. Not to mention there's things like horn modification of the cows, and this would have happened during life of the cow. And we're not quite sure what this means, but it's been hypothesized that it might reflect either a identity of a social group that owned the cow, or even a particular owner, a chief or something like that, and that these cows were then potentially donated as an offering when the ruler of Kerma died. But there's a very strong cattle component within Kerma culture. So we might expect them to be eating more cows potentially. So what does the isotope data actually tell us about Bronze Age diet? Very roughly, I'll put up, in case some of you are familiar with these isotopic data. If we look to the right of the x-axis, you're more likely to get sorghum data. So I've put a picture of a sorghum plant here. And on the other end of the x-axis, we have a picture of wheat. Those data are going to reflect more of a C3-based diet. Again, these are very broad strokes. This doesn't necessarily mean that they were eating sorghum and wheat, but just as kind of a reminder about the data that we're going to be looking at what they mean. And on the y-axis, this is nitrogen. We're going to be looking at, that protein component of the diet. Okay, so these are some previously published data from both Egypt and Nubia. So the red are going to indicate sites that are from Egypt, and the blue are going to indicate sites that are from Nubia. With the one exception of Tombos, which is both red and blue because it's this colonial site that was very kind of Egypto-Nubian, and we know that both were living side by side. So I have that one as a red background with blue marks through it. So we have, a quite a wide variety of dietary patterns being shown here. We'll walk through just very briefly. And here's a map to go with it, just to put it side by side to show you the sites both in Egypt as well as in Nubia. So very broadly speaking, the delta 13C values, the carbon values from Egypt, are more enriched than Nubian values. So in other words, the Egyptians may have been consuming more C3 foods, including wheat and barley, which aligns well with what we know about Egyptian beer culture and economy. So the Nubians, on the other hand, are more variable. There appears to be a mix of C3 and C4 foods at Kerma. This may be because the drought-resistant C4 plants like sorghum and millet grew better in Nubia. However, there's also evidence for significant C3 plant production from Nubia. So this might be the explanation, but there's also alternatives. So another possibility is that there was a cultural preference for sorghum and millet, and this was a point of distinction between Egyptians and Nubians. We'll continue with this hypothesis in just a bit, but nitrogen is markedly similar between Egyptians and Nubians. Both groups were likely consuming moderate degrees of protein. But I should also note, I probably goes without saying, but the standard variation for both Tombos and Kerma is quite large. So there's a lot of variability within both of those populations in particular. Okay, so moving on to another Kerma site to kind of bolster these data. Like Robert said, Dr. Stuart Tyson-Smith and myself have been excavating there since 2015. It is located at the third cataract of the Nile, right between Tombos and Kerma. So it's about 10 kilometers away from Kerma and 5 kilometers away from Tombos. So it's very, very close to both of them. And for this, this is one of the reasons we're calling it a suburban site, is because it was quite close to Kerma. Within walking distance, it would have been a very long walk, but very accessible to the capital city. It was occupied between the ancient Kerma and modern Kerma periods all throughout. So we have a really nice chronological span. Despite a lot of disruption to the cemetery due to modern alluvial mining, actually the majority of the cemetery was intact when we started excavating in 2015. And it displays really, really well preserved, particularly organic material, but also skeletal material. We've been very, very lucky. So I will present on some of the dietary data today. Just to kind of show off some of the cool stuff we're finding. This has nothing to do with diet, but I find it very cool. So we have a a flip-flop on the far right that was from a classic Kerma grave. On the left top, we have a woven bed made of cowhide, a burial bed. On the bottom, this is just a close-up image of some preserved leather that we found, and so well preserved that you can actually see the hand stitching is still very much intact. So yeah, we're very happy with this. A few examples of the graves that we're finding. So the picture on the left is actually a ancient Kerma grave, so 2500 BC. So quite old and yet still really well preserved. The head is noticeably missing. This was due to looting activity, but ancient looting activity. It's really common, unfortunately, at Abu Fatima, but we think that people may have been, maybe stealing necklaces or things like that. But particularly the heads and sometimes the hands are displaced as people looted for grave goods. The dark brown color that you see in the lower body is actually the preserved remains of a leather skirt. So that distinct color difference, that starts right around the waist and continues to the ankles, is bits and pieces of a leather skirt, which is really fantastic. And on the right, it's obviously a preserved foot with a flip-flop or a sandal attached to the foot. And this particular foot is on display at the museum in Khartoum. So yeah. Okay, a quick note on some isotopic obstacles. So several authors, as well as myself and others, have noted the difficulty in actually being successful at extracting carbon and nitrogen from collagen from bone material in Nubia. And I suspect that this is due to the hot and arid environment, and Brenda Baker has suggested the same. Perhaps this is detrimental to the bone and maybe seeps out some of that collagen, such that the bone might look really beautiful, as in some of the images that you saw, but there's actually not much collagen left. So when you go to run the isotope analysis, it's actually doesn't, the values aren't reliable. So in one way, it's beneficial. This environment is beneficial for hair and soft tissue preservation, but not so much for bone preservation. But this has given us the opportunity to actually try to extract dietary information from organic materials such as hair. And so the data that I'm going to show you from Abu Fatima is actually data that was extracted from hair, mainly because I ran skeletal data, collagen, and it was unsuccessful for the most part. I got very, very few reliable results from that. And so I'm relying heavily on hair values. Okay, so to start off, this is the actual values of Abu Fatima hair. However, it's important to note that there is a offset between tissues. So we can't directly compare hair values to bone values. There's a bit of a calculation that you have to do to kind of translate the hair values into something comparable to bone, or closest proxy to bone. And so here we have that new value where the arrow is pointing, and that's the corrected value. So I'm going to use this value from now on, just so you know. But now we can start to kind of interpret these data. So Abu Fatima is arguably even more C4 reliant than Kerma. So it's more towards that sorghum icon. It's around the same nitrogen as many of the Egyptian populations, perhaps slightly lower than Kerma, but not significantly so. There is again, a really even larger standard deviation here. So we're going to take a closer look at some of these data points specifically and kind of tease these apart. So here are the single data points for Abu Fatima, and we can see a couple of outliers, especially that 8A2 that is quite nitrogen enriched, as well as carbon depleted, so looking more like a C4 diet. We question whether or not maybe this, even despite the relatively small sample size, whether this changed through time. But as you can see, the Kerma Ancient, Kerma Middle, and Kerma Classic periods identified by these various colors, there's really no difference in dietary patterns that we can tell at Abu Fatima between time periods. And then we asked about differences between males and females. Again, not significantly so. It looks like both males and females are eating similar foods. And then we've also run strontium isotope analysis to estimate locals from non-locals. And as we just heard from Michele's talk, this is a bit more complicated than just strontium. But these were run several years ago, so all I have is strontium values for these. But perhaps the suggested non-locals based on the strontium data also are kind of all over the place. That 8A2 individual who is an outlier is a non-local. But that, yeah, it's a very small sample size, so it could also just be coincidence. But now let's compare the Abu Fatima data to the Kerma data to really focus on Nubian diet during the Bronze Age. So it does appear that a couple of Abu Fatima individuals are really very closely related to the Kerma data. So there's a few points there that are really overlapping with the Kerma diet. However, on the whole, more Abu Fatima individuals are eating foods that are leaning more towards C4. More of them, I would say most of the individuals are eating a mixed dietary input. So it's a mixture of C3 and C4, which isn't surprising given that most fruits and vegetables and things are C3. But it does suggest that perhaps there's a stronger C4 component to Abu Fatima than Kerma. This could also be one other factor I think to consider is that hair, of course, has a much shorter turnover time than bone. So when we're sampling hair, we're looking at actually, depending on the length of your sample, in this case, it would have been 1 centimeter long. We're only looking at about a month's worth of diet rather than 10 years or 20 years as we do when we're looking at the bone collagen, which is what the Kerma data are based off of. So that's one other variable that we should also think about. So perhaps this 8A2 individual died during a sorghum season. And so that sample that was collected was right around stores when everybody was eating sorghum. Who knows? It's something to think about though. And with the nitrogen, again, they're fairly close together. There's a few individuals in Abu Fatima that maybe are a bit lower in nitrogen. There may be a socioeconomic status thing going on here because Kerma was, of course, a very elite capital city burial, a necropolis of the entire kingdom. So perhaps the individuals that are being sampled from Kerma were of a different status and had access to different foods. And I'm running out of time, but one last thing to note also is that with the nitrogen values, we also need to remember that it doesn't necessarily just mean protein, but it does also reflect the foods that the animals were eating, the foods that the animals were actually consuming. So if they were foddering their cattle on C4 foods, that's also going to influence the isotopic data. So just a quick note on that. Okay, to wrap it up, a few closing points. Abu Fatima seems to be a little bit more C4 and a little less nitrogen enriched. It's possibly associated with status, but we don't know. Definitely more isotopic studies from other Kerma period sites need to be done for comparison purposes. And I would also like to note that hair is a potential viable alternative in cases where bone collagen is not working for people. I've had great success with it. And if you have any questions or want to discuss later, I would love to talk more about it. But for now, thank you very much.
Excellent. Thank you very much, Sarah. That was a wonderful presentation. Thanks for joining us today. We will move on now to our third and final installation for today, which is being provided to us by Dr. Iwona Kożarowska-Ugomakin of the Polish Center of Mediterranean Archaeology, who has a research of particular interest in the bioarchaeology of ancient Egypt and Sudan, focusing on questions around disease and disability, burial practices and mummification, and social stratification. Dr. Kożarowska-Ugomakin has undertaken fieldwork in both Sudan and Egypt, including Meroitic era sites, Theban tombs in Egypt, and most presently at Saqqara. So thank you for joining us today, and please take the floor.
Thank you very much, Robert, and warm welcome to everybody who's present here. Lovely to see you all, and thank you for your time, and hopefully you will enjoy this final talk of this installment. So let me just quickly share my screen with you. Just a sec. Okay. Just to confirm, is everything okay? Can you see my first slide?
Looks good to me.
Yep. Okay, brilliant. Okay, so the topic of my talk today will be on isotopic evidence of an environmental shift at the fall of the Kushite kingdom of Meroe, so approximately 300 BC to 350 CE. So just very briefly about the Meroitic kingdom, I've included some photographs of a few important sites. The Meroitic capital city, currently in ruin, a lovely elephant, as the Meroitic kingdom was known for its elephants, and of course the fantastic pyramids from the royal cemetery at Betroia. So talking about, oh, I've gone too far, sorry. Too far. Right. So talking about climate change and how do we study climate change or evidence of climate change? So, of course, we have a range of evidence coming from geological records, glaciers, archaeological records, historical records, and so on. So there's plenty that we can use and rely on to investigate climate and to find out how climate changed throughout the time. So today, of course, I will be focusing on archaeological records and more specifically on human remains, plus a little bit of faunal remains. So this is what I will be talking about. And just to make you aware, just a few facts on climate change. So over the last 200 years or so, the Sahara has gone through multiple periods of wet and dry climates. So climate change is not something that is happening just recently. The last green period, when the Sahara was green, lovely and blooming, ended approximately 5,000 years ago. So the period of increasing aridification, which started some 5,000 years ago, is essentially the period we live in now. So we're still seeing this increasing aridification, particularly in the region of present-day Sudan. Just to talk briefly about how we can see climate change, which reflects in environmental changes in Nubia, present-day Sudan. So the map shows you more or less the situation at the moment. So we have most of the Sudan covered by the desert. Just the fringes of the present day Sudan are looking green, but southern Sudan is looking very green. And this is because of the southwards shift, climatic or environmental climatic shift. So the climate change goes southwards, so leaving behind in the northern parts, which is Sudan, environment that looks like the top left image. This is actually a photo I've taken in Butana, so a region formerly covered by the Meroitic Kingdom. And presumably the region during the Meroitic period was looking more like the image in the top, in the bottom left. So that would have been a lovely lush savanna, something that we don't see anymore. And how do we know that that land, the Meroitic Kingdom, was essentially environmentally looking like a lush green savanna? We can, for example, look at the evidence that the inhabitants left on monuments. And just to sort of for your attention, I have a few images here, and I'm sure you can recognize the shapes of animals that we normally expect to see in savanna land. So that would be a mighty lion, again, an animal that was quite symbolic in the Meroitic Kingdom, particularly in religion. Giraffes, the second one, and mighty elephants as well. And these animals obviously are long gone from the territorium of the central northern Sudan, but you can still see them, some of them definitely, in the southern, very southern parts in a national park. So this reflects this southward shift of the climate, so going, sort of pushing lush savanna, green savanna, southwards away from the central part of Sudan. Now, talking about climate change, Michele mentioned strontium isotopes being used to investigate migration. Strontium isotope is also used to look at the fluctuation of the Nile. And this study was conducted by Stanley and colleagues, a short contribution to Nile flow failure at the end of the Old Kingdom, Egypt. But they managed to produce this kind of very zigzaggy line as it fluctuated through time, going from pre-dynastic to the period of time that we are most interested in this topic. So that's Greco-Roman and Byzantine periods in Egypt, which corresponds to Meroitic period or Meroitic Kingdom in Nubia. So as you can see, the level of the Nile was quite low at the time, which indicates climatic change that was taking place. Now, just a few quick words about the Meroitic Kingdom. So it was established more or less around 300 BC, following the transfer of the Kushite capital from Napata to Meroe. The kingdom had a great geographical location, which guaranteed a very strong position on the geopolitical arena, but also allowed the country or the kingdom to participate and play a major role in a global trade. They had advantage of a very strong iron industry, which obviously boosted economy. But something that we are particularly interested in today is its agricultural transformations that allow the expansion of new settlements into new geographical and environmental zones. So in other words, the Meroitic Kingdom and people who lived during that time in that particular region were essentially booming and supposedly enjoying really good life. However, what we tend to see from around the sort of first centuries AD, we notice evidence of gradual decline. We just progressed towards the 4th century CE when the Meroitic Kingdom collapsed. The final point for its collapse or fall was the capture of the capital city of Meroe by Axumides around 350 CE. So thinking about the collapse of the Meroitic kingdom and what actually contributed to this once glorious empire to collapse. So obviously a lot of different causative factors have been discussed and are still being discussed, starting with the political instability. Obviously there were different things happening along the borders of the Meroitic kingdom. The border with the Greco-Roman Egypt was not particularly stable, although the political and diplomatic relations were sort of calmed down later on. But then we have Axumides, another growing, very powerful kingdom at the southern border. Another thing was that the economic circumstances slightly changed, and the trade that was previously mainly going through the Meroitic kingdom was redirected to a new route. So the Meroitic kingdom kind of lost in that particular income that was coming through trade. And also with changing political situation in Egypt, there was a kind of decrease in demand for luxury goods that previously were imported from Central Africa. So again, decline in trade, decrease in trade income for the Meroitic kingdom. Finally, with things changing and not being very stable, there are suggestions that the Meroitic kingdom and its population was a little bit troubled. So there was social unrest due to nomadic invasions and migration into the Nile Valley. And finally, recently, some scholars have been pointing out that actually apart or alongside those mostly discussed causative factors, we should actually look into climate change, because quite possibly climate change and environmental changes which took place during the first centuries AD or CE could have been the sort of underlying factors that sort of unsettled everything in the region and led to the collapse of the Meroitic kingdom. So talking about the greatness of the Meroitic kingdom, obviously what we know quite a lot about agricultural transformations, advancements, iron industry. Several great studies are being carried out on those two topics. But we need to also think about what these advancements and transformations and booming industry do to the environment, local environment. So let's think about climate change as a natural process, but also something, but also environmental changes that can be brought by humans who use the environment for their own purposes. So let's think about using the land for agricultural purposes and trying to sort of increase the sort of production, food production. When things are going well and the region is doing well, we can expect a greater number of people coming to that region. So we would see, and when the local groups, communities are doing well, naturally there will be population growth. That would put strain on the local resources. But then thinking again about iron industry, for example, to actually produce iron, we would need fuel, and that fuel would normally come from wood. So thinking about the trees that would be growing locally, that would be obviously cut to produce fuel for to meet the demands of that industry. So there's a lot of things that I think contributed to the environmental changes also not only from as a natural process, but also as a human factor. So all those changes would be reflected in changes in diet and health in the local populations, changes in the environment, which would not sustain expanding agricultural activities, would eventually force people to move away, look for better lands, and that would result potentially in increased movement of people within and between the Meroitic Kingdom and adjacent regions. So what I wanted to find out was really, can we see any evidence of environmental shift using human remains? So I'm a bioarchaeologist, so I study human remains. So as we all know, I'm sure human remains are a wonderful source of information about lots of things, not only about individuals, populational groups, but also about environment. So my hypothesis was climate change as a contributing or even underlying factor to the collapse of the Meroitic Kingdom. So how to investigate it? So I've decided that the best way would be to use dental enamel from post-winning teeth. So we're talking about canine and molars. And I've used sampled dental enamel from individuals coming from altogether 13 archaeological sites. So I've collected samples of dental enamel to conduct isotopic analysis of carbon and oxygen to determine environmental conditions. So this map shows the location of the sites where the samples came from. So as you can see, we really cover quite broadly Nubian region, and we also have some samples from Jebel Moya and as far as the border with, close to the border with Ethiopia. So altogether, we had 64 samples from human individuals. In addition, we had 15 faunal samples because we wanted obviously establish a baseline, isotopic baseline, to really see whether we can see the same values in humans and also in animals, or maybe there were some differences. So here you see the time periods that we have divided our samples into. So we try to really be as sort of precise as possible with our studies in terms of being able potentially to see changes in the ratio, isotopic ratios, to really see whether we can see some patterns of change pointing to climatic or environmental changes. So we have samples that we dated to the early Meroitic period. Meroitic generally, if we could narrow it down, late Meroitic and post-Meroitic. In addition, we had just a few samples from a C group population and also medieval and post-medieval. So that would kind of help us to contextualize our data from the Meroitic and post-Meroitic period. The results I will be presenting today are essentially going to be very soon published in the paper. So this is the reference to the paper that hopefully will be coming out very soon. So no need to take screenshots or be afraid that you miss something out. Everything will be there, everything that I'll be talking about next. So this is just the table to show you the results of our AMS C14 results. So we tried to obtain C14 dates for as many sites as we could, and we pretty much achieved that. So this is obviously to enforce our investigation into environmental shifts. So we needed a precise date for that. And here are the results. So this graph shows you temporal distribution of the C13 and oxygen values in human enamel samples. Statistically significant differences were observed between the 4 meroitic subsets. So we are talking about these 4 groups. Samples from C group and medieval and post-medieval, because they were very small, they were not included in any of the statistical analyses, but they were included in the graphs just to demonstrate how they plot against other meroitic and post-meroitic values. We also noticed that there was a significant difference between the early meroitic data set, which presented the lowest carbon isotopic values, and the subsequent meroitic and post-meroitic subsets. So the significant change in the carbon isotopic values would, in this case, indicate C14 enriched diet in these subsets. And of course, C4 plants are associated with hotter and drier environment. So we also noticed that there were temporal differences in the stable oxygen isotopic values between 3 periods. So we combined, because the samples, early meroitic and meroitic subsamples were very small. So we combined the 2, therefore from 4, now we have 3. So early meroitic and meroitic, then late meroitic and post-meroitic. So differences between those 3 periods are statistically significant. So looking at the temporal distribution of both isotopic values in human enamel samples, we can clearly see a pattern of continuity if we look at between the very early C group and then the early meroitic period. And we also observe a decrease in the oxygen isotopic values between the post-meroitic and medieval, post-medieval periods. Now, interestingly, when we compared the isotopic values from our faunal samples versus human samples, we've noticed some interesting patterns. So overall, both carbon and oxygen isotopic values were on average higher in animals than in humans, which to some extent is not very surprising. But interestingly, we've noticed 2 relatively distinct clusters of bovids. So you can see one cluster here and then another one here. So the second cluster is pretty much covers the same distribution as the ovicaprids. We've noticed that there was a significant difference in the carbon isotopic values between humans and bovids, and also a significant difference in the oxygen isotopic values between humans and both bovids and ovicaprids. So they kind of had slightly, potentially different diet, or they lived in slightly different places. So what do the data tell us? So thinking about the carbon isotopic results. So the data tell us that we can essentially observe an upward shift in the carbon isotopic values following the early meroitic period, which would indicate a progressive dietary shift towards greater consumption of C4 adapted plants such as sorghum, and reduction in consumption of the C3 crops such as wheat and barley. So in other words, we can notice quite clearly there's a shift in the diet. So the people during the later periods, later times within the meroitic period, were consuming more C4 plants. So there was a shift to potentially eating more planting and eating more sorghum and millet, less so wheat and barley. So we then need to think about, if they were eating more of those two crops, then they would plant those crops more frequently. So there was a change in diet was obviously due to a change in agricultural practices. So these findings about the dietary shift corroborate previous isotopic studies that found a marked shift towards a greater consumption of sea plants, and also agricultural biseasonality in the post-Meroitic period. So our results tie in with the observations made by other researchers and published over the past 2 decades or so. So agricultural shifts characterized by intensified cultivation of summer crops, so that's sorghum and millet, could be attributed to the contemporary climate change, also decline in precipitation and increasing aridity in the region. Now, about the oxygen isotopic data. We have observed a clear temporal shift in the oxygen isotopic values between the early Meroitic and late Meroitic, but also post-Meroitic periods, which correlate with the paleoclimatic data that suggest decreased rainfall in Ethiopia and obviously by extension in Sudan during the 1st centuries AD. So temporal differences in the oxygen isotopic values most likely reflect a progressive decline in precipitation due to climate change. So thinking about water availability, if there's less precipitation, then there would be a decrease in the Nile flow and decreased levels of water in the Nile. So it wouldn't be possible for the people to potentially cultivate crops further away from the Nile. So the booming period during the Meroitic kingdom, when the people were able to expand their settlements, move away from the Nile, now that practice would be receding. So they would be coming closer to the Nile due to the lower Nile flow. So what happened to the Meroitic kingdom? So we kind of tend to use certain terms interchangeably. So we talk about fall, we consider gradual collapse, but more recently, we kind of try to stay away from those kind of almost one event things that change geopolitical arena to something that is more gradual, progressive, and long-term. And also try to stay away from sort of negativity, something ends and that's it, thinking more about human adaptation, transformation to new environmental circumstances, but also thinking about political circumstances, as trying to transform from maybe one dominant center of power and kingdom to smaller units, smaller polities. So instead of something that could have happened suddenly, and quite often the fall of the Meroitic kingdom is attributed to the Axumites invasion and the collapse of the capital of Meroe. But I'm not really sure whether one event can really just put a stop to something as big as an empire. There would be different factors contributing. Gradual collapse, so something was happening over a long period of time that eventually led to changes, so most likely transformation rather than a rapid definitive fall. So in conclusion, I would say that our data really strongly support the hypothesis that there was an environmental shift. Climate was progressively changing towards more arid, and it is quite possible that climate and environmental changes were actually an underlying factor that kind of led to other things that happened, and altogether as a package contributed to the end of the Meroitic Kingdom as one unit, and potentially and led to the establishment of smaller polities. So we have isotopic evidence of climate and environmental changes, which likely accelerated during the first centuries CE. Changes in agriculture and diet were brought by changing climate to hotter and drier. So people needed to adapt to new environmental conditions in order to survive. We need to also consider the sort of combined effect of natural environmental changes and also man-made environmental changes, which likely put together increased human mobility. So here we have this, one of the factors, social unrest. And if power was decentralized, so actually the Meroe city was not the central sort of government that governed the whole kingdom, but actually the power was kind of given more to local regions. So individual regions were easily on a path to independence. So that eventually would lead to the formation of regional polities. And as Fuller stated over a decade ago, environmental changes would have exacerbated a trend towards regionalism and also formation of splinter states that commenced with agricultural transformations followed by socio-demographic changes. And indeed, what we see following the sort of end or collapse or fall of the Meroitic Kingdom is the sort of establishment of successor states, smaller polities, which eventually then transformed into very strong 3 Christian kingdoms. So that's our research, which was part of the project Environmental Changes and the Collapse of the Ancient Nubian Kingdom of Meroe in Sudan, which was funded by the European Union's Horizon 2020 research and innovation program. And I would like to obviously take this opportunity very quickly to say thank you to a number of people who had made this research possible. And my special thanks go to my Sudanese colleagues who were wonderful in enabling me to conduct my research in Sudan, very helpful, very supportive throughout. And I very much hope that they are all okay and that we will see each other very soon. Thank you very much.
Okay, wonderful. Thank you very much for the presentation.